Hello, everyone. I'm Nate Truex, and you're listening to the Croccast Podcast.
Hello, everyone. Welcome to the Croccast Podcast. I'm your host, Nate, and today I'm joined by Dr. Stan Trauth, Professor Emeritus of Arkansas State University, with specialization in researching reptile and amphibian reproductive histology. Stan, welcome to the show.
Thank you. Thank you, Nate. Wish we were seeing each other right now, but that's technology for you.
Always frustrating. Yeah. So, do you want us to give us a little bit of introduction about yourself? Tell us a little bit about how you first got into reptiles?
Into reptiles, you're asking me to go back a long way. Now, I was born in St. Louis, moved out of St. Louis when I was 7 to the Arkansas Ozarks, and my parents bought a resort there, and right on the lake, Bull Shoals Lake. And it turns out that the lake itself was down during several years, my early bringing up there, and along the shoreline were collared lizards. And so as a kid, I would run down and chase collared lizards, and if any snakes were available, those too, but mostly collared lizards. So I moved on and went to college at University of Arkansas at Fayetteville, and my major professor, once I did my undergrad there, I got into grad school there, and my major professor suggested, he pointed at me, I gave him some suggestions about animals I'd like to work with, and I said a hellbender and maybe a ground skink or something. Then I said collared lizard, and he pointed at me and said, that's it. So that led me into the reproductive biology of collared lizards. And so that was basically my start in histology, where I published a couple of my papers out of my master's on reproductive anatomy of collared lizards. From there, it just expanded into all the various herps. So I can give you more information. I want you to ask questions too. So go ahead.
So for people who don't know, could you give a brief description of what histology is?
As a study of tissues using a light microscope, and I have 2 light microscopes beside me right now, and about 100 or so slide boxes, or more than that, actually, a couple hundred slide boxes around me of prepared microscope slides. So I was going to give you the grand tour of my lab, and here I was able to move from Arkansas State with materials I had in my lab there, and created a lab in 2 garages here in Morrilton. Histology allows you to examine tissues, not fine structure, but just looking a little closer, up to 1000x. And I have staining dishes, I have microtomes, I actually have an ultratome also to do very thin sections. And so this has stayed with me my entire academic career, doing this type of work. Now, don't get me wrong, I do other stuff too. I do field work, and I, of course, published Amphibians and Reptiles of Arkansas in 2004. First author on that book. And we have a rough draft for a second edition of that book, basically prepared right now, ready to be submitted to the U of A Press. So I do a lot of additional work outside the reproductive anatomy. But to get back to this, this topic, I collect animals. I collected animals. I don't do that anymore, and prepare their gonads or their reproductive tract to be studied with a light microscope. When I was at Arkansas State, I was the director of their EM facility there, electron microscope facility. Had that job for 28 years or so there. So what I did was prepare a lot of tissue for examination, and then I was able to store it in either wax, paraffin wax, or in epoxy resin. And those tissues are still with me right now. And so I can decide on a particular organism to study. Right now it happens to be siren netting eye. The name has changed. Western lesser siren is now the Western siren. So sirens are very unique in many ways. They don't possess hind limbs or girdles. And that's just one aspect. They have gills throughout their lives, external gills. There are so many unique things about sirens that it kept me occupied for about 3 years now. But I took those tissues of sirens that I collected when I was at Arkansas State over a 20-year span and kept them, and then am working on those right now. So the tissues are fascinating. They're new to science. Stuff I'm doing now has no precursor. There's no literature other than maybe some basic comments by some authors about their reproductive anatomy. But other than that, this is all brand new. So very interesting, very fascinating.
Yeah. So you mostly work with like native North American herbs, or do you have any particular groups in general you like to work with?
Well, I work with North American herbs. Everything from lizards, snakes to frogs,
Sirens.
salamanders. I mean, I have used all of the various herb groups in my studies. Excuse me. In my early work, after the collared lizard, I did some salamander work on cloacal anatomy and published a number of papers in Herpetologica, and that was it was now 70s, 80s, a long time ago.
Yeah.
Since then, I've branched out. I work with neurons and turtles. A lot of turtle work. If you go online, you'll see some of my turtle work with alligator snapping turtles, and also I did a paper on mud turtles not too many years ago. So it's the whole shebang. It's all the organisms that you would call herbs I have studied.
Yeah. Okay. So I imagine since North America is temperate, maybe a few extreme other parts you could describe as semi-tropical, you see a lot of variations in the tissue of reproductive tissues in herbs. Is that true?
Well, you can, yes. One must look at an organism and its range and say, well, it may start its breeding season at a different time, as opposed, say, from north to south or south to north, whatever. To see that there are genuine differences in an organism, you're probably not going to find that type of differentiation in tissues at the species level. Now, you can compare species, and that's what I do. Because you're not going in one species, like for example, the prairie race runner to the west of the Mississippi. You have the eastern six-line race runner to the east. Now, that organism was my organism for my dissertation, and I have used tissues from the lizards I collected for maybe about 10 years in studies that I've done. So But to answer your question now, finding variation in a particular species, not really. Okay.
Okay, so there isn't much change, say, from winter to summer then.
Well, yeah, there would be a seasonal variation in the gonadal anatomy due to changes in the reproductive cycle. And I have looked at that. I have not, let's say, I have not gone into sectioning large ovarian follicles versus small ovarian follicles because there's really no need to do that. I look at testicular tissue and find changes in the spermatogenic or testicular cycle. That is interesting, and I'm published on that. So there is variation in the timing of the gonadal activity. So you are correct there in saying there is variation that way. Yeah.
Yeah. So what are some of the changes you see in this gonadal tissue through with the different seasons and different weather?
Well, let's take we can take the collared lizard for example, or actually any North American lizard. You're going to have a seasonal change in the spermatogenic cycle. The production of sperm does not begin immediately when the animals come out of hibernation or brumation, but they have this period of time where they build up toward the release of sperm. So you can go in and look at the spermatogenic tissue, the seminiferous tubules, and follow the stages of spermatocytogenesis, spermiogenesis in a particular lizard. That's going to change, yes. And so we as reproductive anatomists, biologists, have done a lot of that. That was some of the original work beginning in the 50s and 60s. Much of that was done on many of the animals, lizards and snakes. I'm thinking of mainly right now. So that was approached, and so that research was hot for a long time. I got my feet wet doing that, but mostly things have changed. Approaches to seasonal cycles have changed in the approach that students and colleagues are looking at those organisms. They don't look at them histologically like I did back in those days.
Yeah. Just talking to people who breed a lot of North American snakes, they talk about how it is important, especially with the males, to get them somewhat cooled down in order to really trigger spermatogenesis in the spring.
Yeah. There are these the techniques to that herpetoculturists do in order to ensure that the males are ready to release sperm, or if females may be storing sperm. We're talking about snakes now, and that is certainly important in trying to decide when to put the males and females together, or let them go on their own, and they will work things out. And
yeah. So with this changing in tissues and sparking spermatogenesis and all that sort of stuff, is there like one individual key factor in triggering that, or sort of like a plethora of different factors that trigger that? Like, say, is it like just photoperiod that's the main triggering factor, or is it like photoperiod and temperature, or a variety of other things?
Certainly, those two are the primary factors. Of course,
Okay.
age of the animal, young adult versus older adult, that's going to make a difference too.
Yeah.
I found in for example, the prairie race runner, that the older females will yoke up earlier and have 2 clutches at least, and the younger females from the later clutch of an older female, when they mature, they may have only 1 clutch. So, yeah, your body size, the age of the animal is important in evaluating the cycling.
It's interesting you mentioned that because I know a couple of people who've kept crocodilians over the years, and they always tell me that a female crocodilian, like her first clutch or her first few clutches, all have very low fertility, but as she gets older, her fertility increases with each passing year.
Yeah. Yeah, a lot of the, of course, what's affecting American alligators, let's say, now is the endocrine disruptors that we put in the water. And I know you know a lot about that in the work of Lou Gillette down in Florida, where he found that the endocrine disruptors were feminizing the males. And that's not been good. So,
Yeah.
yeah.
Yeah, right now I'm just thinking of a Alex Jones quote when I hear about that sort of stuff.
Yeah.
So
Go ahead.
Go ahead. Go ahead.
I said,
please.
ask.
Yeah. So speaking of that, have you noticed a change over the years with, like, say, some sort of like pollutants or biological? I guess pollutants would be the word, building up in reptile tissues over the years from when you started versus where you are now?
Well, you've got to remember, to do this histology, you're looking at a small sample. And once you do that small sample with a particular organism, you're less apt to want to go back and work that hard just to see if there's something going on that you may have missed or could reinterpret. That's hard work. And so that kind of work, there's only a handful of reproductive histologists in the U.S. And to do this kind of work, it's time consuming, it's laborious, it's you have to have the facilities to do it. So doing a monitoring of change in organ gonadal output or change in its anatomy is very difficult unless you have something set up already where you know, like endocrine disruptors or some pollutant is involved. I found 4 years ago or something like that, some southern leopard frogs. Southern leopard frogs have been studied quite a bit, as you might know, and happened to find a couple, and one in particular whose lower limb, the tibia, had enlarged, and tibiofibula. The fact that there was additional bone there led me to look at normal legs of leopard frogs, and then this particular one that had these abnormalities. And so that was interesting. And then there, of course, there's no way of knowing what caused it, but at least I reported that. The other place it was reported was in India, for example. So I didn't have a whole lot of literature to work on there. These things can happen every once in a while. You'll find an unusual anatomy.
Yeah.
Abnormal anatomy that could have, who knows what the cause is.
Yeah.
Yeah.
So I'm looking through some of your papers right now, and you mentioned earlier snapping turtles. Looking at morphology of, was it wrath glands and snapping turtles?
Yeah, Raskey's gland. Yeah,
Yeah.
I've done 5 papers on Raskey's gland starting in 2009, where Mike Plummer and I did the first really intensive study in recent years on, I say recent years, 2009, on the Raskey's glands. And I did a lot of anatomy. In other words, I had to discover where the glands were and followed their ducts, and that was a very interesting study. Interesting enough to lead me. Mike and I did one other one on box turtles, and then I've done 3 others since then on snapping turtles, alligator snapping turtles, and then on the mud turtle, Mississippi mud turtle. So yeah, those are the musk glands. We call them the scent glands, or whatever you wish to call them. The more recent name is simply named after the 1800s histologist Raskey.
Okay.
And I was going
So you mentioned, sometimes. Go ahead.
I said I was going to show you that
Yeah.
particular. You may have it in front of you. That particular study. So on the cover of the Journal of the Arkansas Academy, pardon me.
Go ahead.
What? I said on the cover of the Journal of the Arkansas Academy in 2001 is a photograph taken from that paper. And it's in color. And you can get all of these papers from the Arkansas Academy. Download the PDFs. They are in color. The plates are in color. You there?
Gland.
Go ahead. You still with me?
Hello?
Yep. You back? You back?
Hey, can you hear me now?
Yes. I lost you for a while. Nate?
Hello, can you hear me? Hello?
Yep. Yeah, can you hear me?
Yeah, I can hear you.
Okay. What happened there? We have a
I don't know what that. Yeah, I guess I don't know. Maybe some just particularly nasty cloud bouncing between the radio tower and us. Who knows?
Okay. Keep going. You're asking about Raskey's glands, and I'll answer anything you wish about those.
Yeah. Yeah, so Raskey's glands, I heard you mention them called scent glands and musk glands as well. So do you mind telling us what role these glands play in a turtle's anatomy and life cycle?
Well, that's the good question. We assume that these are glands are releasing pheromones for a function with either it could be a number of things. It could be reproductive, it could be territorial. We don't know the precise function of these secretions. We know that in all of these aquatic turtles that the secretions are smelly to humans, foul, disgusting, whatever. And I mentioned aquatic turtles. Now, box turtles can be aquatic. They do You think of box turtles being mostly terrestrial, but they do venture into water. And it turns out that box turtles lose their Rathke's glands after a certain age, young. Yeah.
Huh.
No longer. Yeah. They regress or disappear, whatever. But that's why Mike Plummer and I looked at the hatchling of two different species of box turtles, ornate and the three-toed. In the very young animals, there we could see the glands.
Yeah, it's interesting that they lose them so young. I wonder if it's just something tied to maybe being a more aquatic lifestyle. Like, I don't know if Rathke's glands are found in like tortoises, like gopher tortoises, or not.
They are not.
Huh, so
They are not.
it's probably something to do with a more aquatic lifestyle then.
Yeah, one would assume. Yep.
Yeah.
We can only speculate, and of course I don't want to speculate on a topic I don't really have any background in knowing what these secretions, how they are perceived, how they are utilized by the males and females.
Yeah. Well, I guess that heads off my next question, which I was going to ask if they're found in any other groups of reptiles, but I guess they're only found in aquatic turtles then.
That's where they're found.
Yeah.
Sea turtles have. Some of the first done on sea turtles, Rathke's glands and sea turtles, but they're not found in tortoises.
Okay. Interesting. So I guess we now know why musk turtles are called musk turtles.
Pretty much. Stink spots.
Yeah.
Certainly are smelly. And the fact is that alligator snapping turtles are smelly too, and you can smell them when you go up into a creek to trap. When you trap, if you go, I think it's a 2015 paper in the journal, the Arkansas Academy of Science. I published a 20-year study on alligator snapping turtles, and the fact that I was out in trapping them, not that consecutive 20 years, but 5 years. Then I was off for 10, and then another 5. Same population. You can smell the odor of snappers when they're releasing the from the glands.
Yeah.
Smell it in the creeks.
Interesting. Yes, been a well-known stereotype in herpetoculture for years that aquatic turtles in general are just really smelly and really foul up water real quick. So I don't know if that's just purely how much they poop, or maybe there's Rathke's glands also play a part in that as well.
I'm going to guess that the Rathke's glands are not functioning in that regard. Maybe under stress, the animals might release from those glands, but it's usually the poop, as you say, that's fouling the water. If you have them in enclosures where you can't filter the water or clean it continuously.
Yeah. So circling back to something we started off with, you mentioned you were doing work with sirens tissue now.
Yeah.
You want to talk about that at all?
Well, I think I could have shown you pictures, images to explain the story. I really had planned that. Behind me, I have all of the plates I was going to show you, and that's here gone now to do that. As I had mentioned, I put off studying, and well, maybe I didn't mention that. I collected these animals back in the 90s and early 2000s, saved the tissues, and some of the tissues I examined and realized I had no idea what was going on. So I put it off as one might do. And I just 3 years ago, I decided I've got to go ahead and look at these tissues. And man, was I ever astounded that we have a vertebrate, a salamander, that has testicular characteristics similar to higher vertebrates and lower vertebrates.
Huh.
When you think of let's take an average salamander, maybe a spotted salamander. They have what is called cystic spermatogenesis, and not just cystic. Let me rephrase: cystic lobular spermatogenesis. If you go to higher vertebrates, reptiles, birds, and mammals, you have acystic tubular spermatogenesis. So there's a deep contrast between fishes and amphibians and reptiles, birds, and mammals on how you look at the testes. In other words, you have cysts in fishes and amphibians, and tubules in reptiles, birds, and mammals. Okay, the siren is acystic, which is similar to higher vertebrates. Nothing like all the other salamanders, which are all cystic. No, not even close. They are also lobular, which is a characteristic of lower vertebrates, fishes, and amphibians. So they have a characteristic there, but the production of sperm is absolutely different. Absolutely different. If you move to higher vertebrates, where you have acystic, we have seminiferous tubules that undergo spermatogenesis. The siren is also acystic, but it's not tubular, it's lobular.
Huh.
Now, with that stated, there are so many differences in what's going on in the histology that someday there will be books citing my work. They have to, because thus far you have in all textbooks you'll have cystic spermatogenesis in the lower vertebrates and acystic spermatogenesis in the higher vertebrates. Well, sirens are acystic like higher, but lobular like lower. There you go. And,
Okay.
and that's the beginning of the story.
That's only the beginning. Wow.
I just yesterday put 2 plates together. A plate is when you put images together into a figure. So that's called a plate or an image. That is just astounding. Astounding. And I state this, and how many papers have I published on this? Okay, I published one 2 years ago, and another one's coming out, it's in press, coming out in 2023. I'm submitting another one this spring, so that'll be a paper coming out in 2024. And I'm thinking about putting a second paper in that 2024 one, which is what I'm working on, which I just mentioned right now, as of yesterday. So it's so beyond remarkable that you don't know how to emphasize it. It's totally different from either. Okay, let's take, for example, tubular spermatogenesis in higher vertebrates. You have a basal membrane, and then you have primary spermatogonia and then secondary spermatogonia. This is in layers now. Then primary spermatocytes and secondary spermatocytes. spermatids, and then sperm. That's going from the basal membrane out toward the lumen of the tubule. In and in higher vertebrates, Sertoli cells help feed those sperm. The sperm heads are embedded in Sertoli cells. These are called nurse cells. Totally different, absolutely totally different in sirens. They are acystic, but nothing like that. And so they're in an order of their own, and I've stated that before, as others have. They need to be separated from salamanders because there's so many differences.
Yeah.
That they need to be in their own order. So that's my say about that.
Yeah. So, can you go over the differences between cystic and acystic spermatogenesis?
Yeah, let's take cystic. Well, in the lobules, the cysts have one stage of spermatogenesis. So in the testis, you see these cysts. These are circular structures, okay? And in each of those, you have all primary spermatocytes. And then another cyst, all secondary spermatocytes. And then another cyst, all spermatids. And that's what cystic is all about. They go, and those cysts then, and within those cysts, they mature and then are passed out the lobule through a duct system. All right. So those are cysts of similar cells. If you go to acystic, higher vertebrates, I just described that. You have a tubule, and along the tubule's edge, margin, you have all these stages side by side, back to back. You remember from your biology from college, maybe you didn't have it, maybe you did. But anyway, you go from the basement membrane, where you have the spermatogonia, all the way out to where you have sperm in the interior of the tubule. Okay. In sirens, it's acystic, but it's nothing like higher vertebrates. Nothing. It's nothing like lower vertebrates. Nothing. It is so different that one wonders why it took 20 years to study them. Because I didn't know what I was looking at primarily, and at that time I had too many other things going on. I needed time to reflect and examine, and then come to some conclusions, and they're just amazing.
Yeah,
Sirens. Yeah. Now I've also done pseudobranchus. I'm sure you're familiar with pseudobranchus, which is the dwarf siren. And in 2017, I published a paper in Copia with several other authors, me being the senior author, where I studied the testes of the dwarf siren. And they are very similar, but not exactly like the Western siren. But I published that. So there's a paper out there talking about acystic lobular spermatogenesis already. I published it, but it is so. I didn't get to do a thorough job analysis of that because I had a limited number of animals to study. And when I looked at the siren, it allowed me to go into more detail because I collected quite a few sirens in order to try to understand what was going on. And you can find that study in the Journal of the Arkansas Academy of Science. You may have seen it already. It's there a couple years ago.
okay.
So anyway, I could talk all day about sirens. I'm looking at their sperm now using the electron microscope. So I'm looking at using the scanning electron microscope, which I had at Arkansas State, and the transmission electron microscope, which I also had at Arkansas State. So I have lots of images to work with there. Lots of them. Yeah. And they are also fascinating.
So, is their sperm structure as different from other salamanders as their testicular structure is?
Absolutely. So different, so amazingly different that people will be astounded. Scientists will be astounded by the differences. Astounded. For example, I gave you an example of the nurse cells, the Sertoli cells, where the sperm heads are embedded in the Sertoli cell cytoplasm. And you can see that in any textbook. The sperm are embedded there. Nothing like that. There's nothing like that. The Sertoli cells are not even involved once you get to the spermatid level. The spermatids are out there by themselves inside the lumen. There's nothing to do with the Sertoli cells anymore. Sertoli cells did their job early on. And so you have these cells, which are extraordinary. So you can tell I can talk about sirens all day. I was hoping to show you these, but it's really a shame. The unusual nature of these cells.
Yeah. We'll tell you what. When I do the social media post about this episode to announce this publication, if you want, I can also show some of the pictures of those as well.
You can of which one?
When I make the social media post announcing when this episode is published, if you want, I could also show some of the pictures as well if you want.
Yeah, you can do that. You can go online and capture it. You know, once those PDFs are out there, you can grab those and make plates, you know, figures and whatever. You can show those. Yeah, the next paper will come out in April. The next in this series, and it's on spermiogenesis, and the spermiogenesis paper is just a killer. I mean, it really is. It has spermatid. You know what a spermatid is? It's the stage before a mature sperm. If you go online, you look at the mature sperm, it's got a head, it's got a midpiece, and it's got a tail. All right. If you look at the siren sperm head, it has 8 folds of the plasma membrane that go into the formation or involved with the formation of the biflagellate sperm tail. Biflagellated sperm. This, you know, and they are unique. Biflagellation in the siren in the sirenids is unique. There are some neurons, a couple, that have 2 flagellae, but not entirely along the entire sperm length like it is in sirens. And I was fortunate to be able to describe those in my own words. And they are called pleated. It's a pleated plasma membrane. It's a membrane that has so many folds in it that have everything to do with the formation of the flagellae, the 2 flagella. It's hard for me to talk about.
Yeah, that's interesting.
They're so fascinating. It's hard to talk about.
Yeah, going into this conversation, I didn't know there was a group of animals that had sperm with 2 flagella.
Which one?
The sirens.
Okay, ask your question again. I didn't hear that.
I was going to say before this conversation, I didn't even know there was a group of animals that had sperm with 2 flagella.
Yeah, yeah, yeah. And many people who don't talk about, listen to, study this type of stuff would not know that. That's okay. I can list half a dozen things about sirens that are different from regular, we call them regular salamanders, typical salamanders. I took the spotted salamander as an example. Sirens, female sirens have no spermatheca, a sperm storage gland. They don't have that. They don't have any glands in the pelvic region. The males, we're not certain about how they fertilize. I think they maybe by cloacal apposition, maybe, and with females, we're not certain. Males, they don't have glands. Cloacal glands. It's extraordinary stuff. What it is with this particular animal, and so I was fortunate to be able to collect these and have the time now, opportunity to thoughtfully think about. That's redundant, I guess. Be pensive about what I'm looking at. I have colleagues that I can share this with, and I have shared it with some and have presented it at a national meeting before in some regard. But what I'm doing now was not presented at a national meeting.
Okay.
I didn't know it at that time. Years ago, 2015, 14, I may have published or presented it. ASIH, JMIH. So
Yeah. Okay. Yeah.
I'm also looking at anurans and looking at urine urogenital anatomy. Very, very interesting. I've looked at wood frogs. I'm looking at just published or put together a paper on Cajun chorus frogs. I've put together a paper on crawfish frogs, which is going to be, it's in press, going to be published this spring. So I'm doing other organisms available to me, but the siren is dominating right now.
Yeah. Yeah. I do want to get to some other organisms as well, but I haven't had a chance to talk about sirens yet on this show, so let's go ahead and keep talking about sirens.
Well, all you have to do is go to Journal of Arkansas Academy of Science, and I think it was 2022 paper came out. I should know that, but I have the paper in front of me, but it's the draft of it. It's not the no, it's my draft. It's not the published PDF. And you can get a lot of information from that paper. But the next two papers that I'll publish are actually three papers. Are going to really be something. Well, it looks like I've lost my image of me I'm back to my email. I guess we get tired of having me as an image there. That's too bad we couldn't have linked up. And maybe that's, it's Google Meet. My computer downstairs here in my lab. I have two computers down here that I work with. The other one is not online. But that's the problem. It was Google Meet versus Zoom, I guess. But I've Zoomed before. I think it's just what the web browsers want to do is change to what they want you to have. And they just go ahead and do it, and you don't have much of a choice. But you ought to look into that Google Meet
Yeah.
and see if you can get that also along with Zoom. So, any other questions you might have?
Well, this podcast is called the Crockcast podcast, so I do it to feel obligated to ask about crocodilians at all if you've done any research with them.
Well, I told you that I did do some kidney work for my grad student who got his PhD recently, and I guess it was University of Central Florida. He's now at the University of Missouri on a postdoc. Anyway, I did a large number of kidneys for him, and he was looking at kidney tubules and looking at variation in the young alligators that he had. And I don't know whether they were treated or just exactly what they had done with them. All I did was do the histology. I didn't ask questions. So I did a good 100 of them, I guess.
You basically did all the grunt work.
Well, they had to get out in the field to get the animals, I guess. But I didn't mind doing. I didn't mind doing that because Johnny was a very good, very excellent student, and I was willing to do that to save him time, so he could look at other things more molecular related with the animal. So I did it. I said I would do it, so I did it. And so that's, I started on the testicular anatomy in alligators a long time ago, but realized I didn't have enough tissue and I didn't have it prepped the way I wanted in order to make it a meaningful study. So I just dropped it, let others do it, and others did it. My colleagues, I've got 3 histo colleagues that were involved with that study years ago. Yeah.
Okay. Yeah.
You're calling. Oh, you are in what city are you in?
I'm in Ohio.
Yeah, what city? Ohio.
You want to know it. It's just some little farm town in the middle of nowhere.
Okay, there's a nowhere school up. I say nowhere. Hold on. Let me get my book out here. Find out where he. There's a little university in northwest. Name some of the smaller universities in northwest Ohio.
Here,
South of
Findlay, Heidelberg.
Hamnet, Findlay.
Okay.
That's it. Where I have a colleague at Findlay. Then I have a colleague at University of Indianapolis. And I have a colleague at Southeast Missouri State. So those 3 individuals. I had a colleague that passed away 4 years ago. He was really the ramrod of about everything we were doing at that time. But those 3 individuals are histoherpetologists. If you're wanting to know the term that I apply to myself and to these others, histoherpetologists.
All right.
So that gives you an idea. Anyway, I don't have to give you the names of these individuals, but you could pretty much find out by just looking in the histology of just about any lizards. Or let me give you one example, and this will lead you. Here is a book I'm holding in my hand. Reproductive Biology and Phylogeny of Lizards and Tuatara.
Huh.
Volume edited by 3 authors, and I'm the 3rd author.
Okay.
You can just go online. You can pick that up.
Yeah.
And see who the people are.
Okay.
Reproductive Biology and Phylogeny of Lizards and Tuatara. That's the name. That's the title. And this dates back to 2014. So this book came out. It's a compilation of a number of scientists, a good number of them. And let's see how many. I'm looking right now. I don't keep this on the top of my head. 17 papers. And that gives you an idea of some of the work that has been done. You there?
Okay. Yeah, I'm still
Okay.
here. Yeah.
Well,
So
go ahead.
No, you go ahead.
Well, I was going to ask about you. I didn't pursue you online, other than maybe looking at your website for a minute. But tell me a little bit about yourself, so I can have a little bit of knowledge there. Are you avid herper? Are you a?
I do a little herping on the side when I get a chance. Right now, weather isn't exactly the best for herping at the moment.
No, unless you're going after salamanders and creeks. You are a herpetologist. How many snakes do you keep?
Oh, about a dozen at the moment, plus a juvenile rhino iguana.
Nice. So let's see. That tells me a lot right there.
Yeah, but I've also done some work with a couple of different reptile parks in the past. Most notably, Crocodile Encounter in Ankleton, Texas.
Okay.
And when I was in college, I was working for Dr. Chris Carmichael at his Serpentarium.
What university?
Bob Jones University.
Okay. Yep. Well, this gives me a little background too. Really, I'm disappointed I couldn't show you all these things because you would be totally flabbergasted by the images. I had a bunch of images to show you because it's been easy for me to do. So when these papers come out, you can put this together and maybe have a better understanding of what this histoherpetologist does.
Yeah. So we've talked a lot about sirens, but are there any other groups that you've worked with that you would say is your favorite you've worked with?
Yeah, lizards.
Any particular lizards, or like eastern collars, or?
Well, published X number of papers on college lizards. Race runners, I probably published 20 papers on 6-line race runners / prairie race runners. Those are the most papers I've published on in those 2 animals. But I've got papers on fence lizards and skinks. I know you probably get Herp Review, and look up my name there and see what I published in the last few years there on various lizards. But yeah, those are my favorites, I guess you could call them.
Yeah. Are there any particular groups you haven't looked at that you want to look at?
There's one animal I'd like to look at, and that's the tuatara. But I I asked for tuatara from several museums. They either didn't have them or they can't give them out. Then I asked a research biologist from New Zealand, and she wouldn't let me have one. She could say I could come down there and do the work down there, but she wouldn't send me a 10 millimeter strip of tissue from one of the organisms. And I sort of, I wish I had that, because it would help me out in a major paper on urogenital anatomy in lizards. It would help me understand the information I have. But I've gotten tissue on urogenital anatomy for a large number of lizards. I think about 45 from various museums throughout the country. And I'm sitting on that right now. Among other things I'm sitting on.
All right. And do you also keep any herps at home, or
I have two smallmouth salamanders.
Solid choice. Solid choice.
I don't keep. I just don't keep the animals. I would love to have a snake and keep up with a snake, but I don't keep animals. I have two salamanders in a terrarium, and they've been with me for about 4 or 5 years now. Give them earthworms, they do fine. So I don't keep live animals, basically. I don't kill animals anymore, other than maybe 1 or 2, 3 a year. At one time, I was known as Dr. Death at ASU, because everything I collected, I saved into the museum, which kindly they named after me after I left. But the fact is that I had to have tissue to study, and we had to know about distributions in Arkansas. And so the Amphibians and Reptiles of Arkansas 2004 would be something you might be interested in, and just looking at. Go online, read about the book, see some of the images there. I don't know what they offer today, but I don't deliberately kill anymore, although I probably had something to do with killing 25 to 30,000 animals and putting tags on them and putting them in a museum so somebody else can study them other than me when I'm gone.
Yeah. Do you also do any herping in your free time?
My feet don't allow me to do it anymore. I, for years, herped, needless to say. And I reached a point where climbing over mountains, turning rocks, looking for animals, my feet just simply gave out. I say it's foot neuropathy. It's not related to diabetes, because I don't have that. But my feet just got to where they hurt all the time. So I knew what the problem was. I still walk. I try to walk 2 miles a day. But to go out and herp, I love to do it. But I am 75, and I'm reaching that point where you can't get out in the wild like you used to. It's that simple. And so I accept that and work in my lab here, where I have tons of work to do.
Yeah. Have you mostly just
Go ahead.
Have you mostly just herped in Arkansas, or have you ever done any dedicated herping trips?
Well, to go elsewhere, you need permits. And I've herped in Texas, and Oklahoma, Missouri, well, throughout the Southeast. into Arizona a little bit. I mean, I don't go deliberately because the permits required today. But years ago, when the permits were not as important to state federal agencies, I watched it in New Mexico and Texas, and for race runners go, I've collected in 25 states. So that gives you an idea about where I've gotten race runners. So not all of those under permit. Most of them were not. Alabama, Georgia, North Carolina, South Carolina, Florida, Louisiana, Mississippi, Tennessee, Arkansas, Louisiana, Texas, Kansas, Oklahoma. I've gone all of those places. And while I was looking for those race runners, I did collect other animals. Yeah. That was back when I was younger.
Yeah.
Back when I was your age.
Yeah. Yeah. Let's see here. So what have been like your favorite type of snakes to study?
Usually the smaller the snake, the better, because I can do the reproductive anatomy without having a problem based on size. Which ones have I done a lot on? In this book, let me mention this other book. If you go into another reproductive biology and phylogeny book, the title is Reproductive Phylogeny and Biology of Snakes. And in that book, I have a chapter. And the title of that chapter is Male Urogenital Ducts and Cloacal Anatomy. And in that chapter, I examined 35 snakes. It is a unique study in and of itself. I did it with my now deceased colleague, Dave Seaver. Now, if you mention Dave Seaver in Ohio, or look up Dave Seaver in Ohio, you get a lot of hits. I can tell you right now. Anyway, in that book, I examined mostly colubrids, but not all. I did some protelids and, but mostly colubrids.
Okay.
Favorite snake? Maybe rat snake.
Can't go wrong with a rat snake.
Yeah, there you go. But in that chapter in that book, I just mentioned one of the emphasis was on, we'll call it a Western rat snake. Burbrink would like that, Western rat snake.
Yeah.
But in that chapter, every small, mostly small. I did water snakes too. Yeah.
Good old Nerodia. Probably your hands probably stank by the end of the day working with those things.
Yeah, bitten and stunk. Right. So I'm still working. I got a colleague who was at the Carnegie, who I will work with next year on water snakes.
Yeah.
So I'm not water snakes yet. A very fascinating study there.
Yeah.
So I'm up to my gills in work, and that keeps me busy, keeps me out of trouble.
Fair enough.
Keeps me out of the woods to go and enjoy the herping. I mean, I've done everything from scuba diving for Ozark hellbenders to trapping for alligator snapping turtles to going into mine shafts after Western slimy salamanders. Did that for 17 years. You might look up my name and Sir David Attenborough. Yeah, I think you probably heard of him.
Yeah, I have. Who hasn't? It's a better question.
Yeah, right. Well, a neighbor of mine hadn't heard of him the other day. I just happened to know. But anyway, you'll find maybe a picture with me with Sir David. And you might also see some pictures of me with Mike Rowe from Dirty Jobs. So
Okay.
there are the internet is great. I had a great website at one time when I was at ASU. As soon as I left, 6 months later, they crashed it. I mean, he's retired. Why do we need his website here? Gone.
Yeah.
I've never developed another website. No need. Retired. You go. But anyway, I did some work with both of those individuals. Had a chance to do some work with Pixar also, but that didn't. It fell through when the movie Newt was canceled by Pixar.
Oh, I heard of that.
Yeah,
Yeah.
I was one of the consultants. Not causing it to be canceled, but I did work with them and have pictures of being in the field with a number of those individuals. So I've had a varied experience academically and have had the opportunity to be with some noted, famous Sir David people.
Yeah.
Mike Rowe's fairly famous too, I guess.
Yeah.
So I can't complain about what I've done, and I've enjoyed presenting all this to you today.
I certainly enjoyed listening to it. So thanks for coming on the show.
Yeah, and let me know if you need any additional info. But you can check me out. I mean, obviously you have or plan to online and pick up any additional info. But I think I've laid out a lot of what I'm doing now for you with respect to reproductive biology perks. So there you go.
Okay.
And I enjoy doing this, by the way.
I'm glad to hear it. I'm glad to hear I wasn't pestering you with my questions.
Anything else?
No, that's about it.
Okay.
All right.
Well,
Thanks for coming.
Hey, appreciate. Get back with me if you need any additional info.
I will. Thank you. Thanks for coming on the show.
All right. All right. Bye-bye.